Welcome to Climbing the Immune Ladder, Steps for Success in Science, an iiSIAR podcast, the show where we explore inspiring experiences in science and the untold stories shaping our community. Today, we'll be your host. My name is Chiara Testini. I am a researcher in immunophysiology at Uppsala University in Sweden.
And I am Janine Lückgen, senior postdoc in immune-mediated inflammatory diseases.
In each episode, we will dive deep into conversation with experts on innate immunity in sterile inflammation, autoimmunity, and their resolution, to bring you insight, ideas, and inspiration you won't hear anywhere else. Whether you are tuning in during your morning coffee or while you're doing your lab bench work, get ready to learn, laugh, and see the world from a new perspective. Let's get started. Today is Steffen Jung, Professor of Immunology at the Weizmann Institute in Israel. Thanks for joining us and welcome to our podcast. Could you please tell us a bit more about your line of research?
So, yes, hello everybody. So my lab is interested in mononuclear phagocytes. So this is a family of innate immune cells comprising dendritic cells, macrophages, and monocytes. And we are studying these cells in vivo context, looking at their development and also the function in different physiological settings, both in health but also in pathology. And let's say one of the strengths of the laboratory has been the development of tools, because we have realized that the tools we have available often have limitations. And so we essentially keep on trying to improve these, and by developing these new approaches, then also to gain new insights. We focus specifically on the gut, on the brain, more recently on the lung, but we are also very much interested in monocytes and the relation of monocytes to the tissue resonance sets. And maybe just as a last point to add, when I started my lab, which is about 20 years ago, we really knew very little about the organization of this field. And essentially, it was a kind of a coincidence, both part of our tools which we developed, but also time-wise, that the field really has exploded, and we have really understood many interesting things. And this has become really a major focus, actually, recently.
And can you share with us what is your trajectory in science?
I started at the University of Bonn in Germany, did my diploma there, and essentially what happened is that during my studies, we had a seminar, a journal club, on a very famous paper from Susumu Tonegawa on the VDJ recombination. And this paper just blew me away, because it was, I think, until today, it's one of the most amazing breakthroughs in immunology. So since at that time Bonn was really not so developed in this area, I moved to Cologne, to the Institute of Cologne, which was headed by Klaus Rajewski back then in these days, and focused from then on immunology. And I did my PhD, I finished in 1992, and then moved to Israel for postdoc, which was interesting, but maybe not too successful. So I did another postdoc at NYU at the Skirball Institute with Dan Littman. And then in 2002, I moved back to Israel and took the position as a faculty in the Weizmann Institute. And that's where I'm actually the last 3 years. So I worked actually during my scientific career. I initially worked on B cells. Cologne was the mecca of B cells. Then I studied T cell signaling, figuring out that signaling is not my cup of coffee. And then I moved to New York, and there I developed these tools which allowed us to look into mononuclear phagocytes, macrophages, microglia, and so on. And this was then actually what I followed up here in the Weizmann Institute. And built the basis for my own work, actually, for the lab.
Did you have any pivotal moment that shaped your journey, either planned or serendipitous?
So I focused early on, because this was also the expertise in Cologne, in the generation of gene-targeted mice. This was actually, we talk about the 90s, where the technology with the cell transgenesis or so was established, and we were pioneers back then in Cologne. And I think this gave me, let's say, I understood that the study of in vivo aspects is very important. And this is also what I then continued during my postdoc. And I generated the mice which carry the reporter gene. So we are talking also, again, I think it's very important to see the timeline in this aspect. So the end of the 90s, intravital imaging was coming, imaging approaches were improving, and we generated a mouse which had a GFP reporter insertion under CX3CR1 promoter. So it's a promoter which we didn't know so much about, but which turned out to be very broadly expressed in mononuclear phagocytes. When we studied these mice, we saw, for instance, in the intestine, millions of cells sitting in the lamina propria. And I remember when we first showed this to our colleagues, they didn't believe it, that these are macrophages. This was just really shocking. But we confirmed this afterwards. And these reporter animals also allowed us now to do adoptive transfers, follow trace cells, and to look at microglia. And other people actually use this to perform intravital microscopy studies in microglia, showing live movement of microglia, which was also, again, around the millennium switch. It was the time when we started to appreciate dynamics of cells in the tissue context. And I would say these mice, for instance, were kind of a pivotal moment, because they put me on the track, and I stayed actually with this promoter, the CX3CR1 promoter, for quite some time. We use it now in different aspects, but we still stay with it. Interestingly, maybe as a side note, this paper, which was these mice, we did a knockout of this gene. The mice didn't have a phenotype. So we essentially had a very hard time to publish these mice. Eventually, it was published in MCB in a section for transgenic mice without phenotype. But it may be interesting for the listeners. This paper is now cited, I think, 800 times or so. It has an amazing high citation index. And so it was something, you could never predict how things go. Yes. So this was one thing which I developed. Maybe I add here, at the same time in my postdoc, I also developed a technology to ablate cells in the in vivo context, which is a diphtheria toxin-based approach. So this was done together with Richard Lang, based on the fact that mice are resistant to diphtheria toxin because they don't have a high affinity receptor for this bacterial toxin. And what we did is we introduced the primate receptor into mice and could thereby confer sensitivity to the toxin to specific cells. We used it for dendritic cells, and we're together with the Japanese group, the first ones which introduced in vivo cell ablation, actually, in a good way. This approach is now widely used also for non-immune cells, but non-immune cells and so on. And again, we stayed with this, although we moved a little bit out of cell ablation because of the potential collateral damage. So we focus more on gene ablations these days.
You have already mentioned several discovery and projects that impacted your field. Is there one that you are most proud of?
So I think it's probably fair to say that the main or one major contribution of my laboratory is the development of new tools, which we use in our laboratory, but which we also share with others. And I think it's important to notice that generation of tools is something, it's a major investment, because we know all our tools, all our models which we develop have shortcomings. And in order to use them in the right way, we have to understand how they work. And it's a major investment actually to define what are the shortcomings. Often it comes only with time, but this is something which we really pushed. And in a way, a contribution of our lab to the scientific community is actually that we shared these animals, something which I learned from Dan Littman, who also had this strategy always. Not everybody has it, but I think it's very important because in the end, it's, the scientific endeavor is a global effort, and we do this together. And so we profit if people use our tools because we get new insights, and it's all about sharing.
I think I talk about from the side of all the community that actually this is really something great that you can share all these tools with us, not only because I'm personally using it in my everyday laboratory life, but many scientists use it. So the fact that you share this tool is great globally for the whole scientific community and research. And thank you for doing that. What is your process for identify this high impact research question, or what drive you to one tools more than the other one? How do you reach the hypothesis idea? What is the process?
I think, I mean, one approach at least is to look for the gaps in our knowledge, right? I mean, being in the field for some almost 30 years, I think I've learned that there are certain issues, people tend to avoid certain issues which are hard to attack, technologically attack. And with the development of tools again, and the progress we have, for instance, in the establishment of CRISPR-Cas9, in the readout methods, single-cell transcriptomics, other omics, we have come actually, so we have such a nice repertoire of tools that we can address really any question we want. So our approach usually, there's two ways. One of them is to identify the gaps and try to understand basics. For instance, what's the difference between a dendritic cell and a macrophage? Why are dendritic cells so superior in priming naive T cells? I think it's a very basic question, and I don't think anybody can answer it. There have been proposed, there's ideas around it, why this could be, but there is no real explanation. And another case, for instance, autoimmunity. I think, since I studied immunology, people have tried very hard to understand or the basis of autoimmunity. And of course, there is progress, but there's still huge gaps in this area. And I'm sure there are things we are missing, really, probably major aspects which have to be addressed. This is one. So this is more, let's say, the rational approach. The other one is I believe very strong in serendipity and in observing the data. So I think the generation of data is very important, and we often try somehow, we look at them, we have certain expectations, and we get a result. But I think it's very important to look at these data which are kind of contradicting our expectations, which often are handled as outliers or so, and often actually there lies something new, and one has to observe it. Fortunately, now in my position, I have the resources that I can actually follow up on such chance observations, at least to some extent, and sometimes they're interesting stories. So, for instance, I can just, it's a kind of an anecdote almost, but we are interested in mononuclear phagocytes, but recently we entered into the field of fungi and yeasts because we made a chance observation that we had animals which we wanted to colonize with Candida albicans to study Th17 cells and so on. And we were unable to colonize the mice. And first, of course, I told my postdoc, this is, just try it again, you do something wrong. And in the end, actually, what we found is that the animals which we tried to colonize had another fungus in their gut microbiome. And we identified the fungus, we sequenced it, and we found that it belongs to another species, Kazakhstania. And we are now actually following up on this and trying to understand what is the basis for this competition between these two fungi. It's something which is specific for the gut, but very robust. So essentially, mice which have Kazakhstania fungus are protected from candidiasis and fatal pathology, actually, for instance, during immunosuppression. So it's something, again, if we wouldn't have followed this up, it was a completely new finding. It's actually now confirmed by other labs, which is good. But it just means keep your eyes open for such things and follow it up.
I suppose that is also a good approach on how to push forward in challenges moment. What can you tell us about strategy that help you to push through in toughest scientific challenges?
I think the key word here is persistence. I mean, as a scientist, we have daily frustrations and we have many, many experiments which don't work, but it requires persistence not to give up. To the extent, of course, one has to be careful because at some point, if one is too stubborn, then one can spend the whole career on a dead end. I think trying to tackle certain challenges by different technological approaches and using the best tools available actually to address the questions, I think nowadays is very important. And like I said, there has been a huge progress. And if it takes a collaborator who can help to do it state of the art, it's probably worth to do it.
And can you share with us any crucial decision or opportunity you had that significantly advanced your career?
Not sure these were, decision for me. For instance, a very critical decision was to come to the Weizmann Institute because the institute really gave me the opportunity to develop my research program. Like I'm saying, well, I said, we are very much focused on in vivo work, on work with animal models, generation of new mutants, and for this you have to have the setup. And the institute is really there, really good. We also, fortunately, as opposed to Europe and other places, our animal regulations are different. We have regulations, and it's also very important that they are there. And we care about the being of the animals, but it is still reasonable, and it allows us actually to move also relatively fast. And in addition, the institute has a very nice collegial spirit. So we have a lot of interdisciplinary research. So I think for me, definitely this was the right move. And I think it's important to be in an environment which is also stimulating.
As a PI, you have many responsibilities. How do you evaluate and seize new opportunities while managing existing commitments?
So essentially, like I said, fortunately, throughout the years, we could secure funding, which is, of course, important. And this funding allowed us actually to pursue new experiment aside, right? The way this usually works is that you apply for funding for defined projects, which of course you have to deliver towards the funding agency. On the other hand, these funds also allow you then to develop new research lines, generate preliminary data with which you can then apply for additional funding. And I think it's just a continuum like this. So there's different approaches. Some laboratories are very much focused on essentially one molecule or one cell type and rather narrow, which allows them to dig very deep. Our approach is that we go actually a little bit broader, which has its risks, but which makes it also, from my side, more interesting sometimes. And so one has to keep this balance between these approaches.
And you mentioned that the crucial decision was to go to a Weizmann Institute. But looking back, were there also career moves that you initially doubted and that turned out to be the right or the wrong choice?
We took at some point, or I took the decision to focus on these in vivo models and the study of mice. And of course, this somehow is connected with certain limitations because these models, first, it's very expensive to work in this area. Second, it's a very time consuming. Especially lately, we study aging, or we entered into aging, and this becomes really a nightmare. It's very hard to combine even the time of a PhD thesis with aging studies. I learned this. Sometimes I thought I should have stayed in vitro and worked on the cell signaling, but it's just didn't give me the kick. So I don't know. It was an interesting development, actually. I think I didn't know this before, but we have come to realize that macrophages, for instance, in vitro and in vivo are completely different. What we have in vivo is tissue macrophages in the brain, in the gut, in the liver, wherever, which are very different and fulfill tissue-specific functions. And this is something which cannot be studied in vitro. And so I think in retrospect, our approach was also then justified, not only because we think Alberto Mantovani said, like, in vivo veritas, right? You could argue, this is what you want to study. But it also makes sense because it's the physiological context which is so important. But it's you pay a price for this. Yes.
You have been talking about some traits that you use, like sharing, collaborating, having support from the institute and from the colleague that really shape your research. And these are all part of non-scientific skill that you develop over the time during your career progression, I suppose. Do you have any other non-scientific skill that have been most critical to your career progression, and how did you develop them?
I think it's very important to keep the curiosity and to keep also a wide angle. I tell my students to go to seminar. I think it's fair to say I've never been in a seminar where I haven't learned something. Sometimes it's it's incremental, but you can never predict what's happening. So I encourage them, and I also try to do it myself, to as many seminars and get exposure and exchange with people, particularly not in my field, because if it's not in my field, we know essentially, we often think we know the questions and we know sometimes even the answers, but it's very good to look beyond the fence and out of the box, because there are really interesting things happening, and I think this is what I would also recommend everybody. We are much too narrow. This is our point. We tend to be, because we of course have our daily chores. We are too narrow in our exposure, and we should start this.
Do you stay current with scientific advancement and constantly foster your professional growth?
I try to attend critical meetings and have scientific exchange. And it's hard to follow current literature because of the sheer amount. But I think trying to be connected is the most important issue here, and see what's following up. And nowadays it's easier because you no longer have to go to the library, so you can get alerts, and I follow the major journals, and then of course I think it's important to do this as a group effort. So I also encourage my team to keep track of new developments, and they share with me what's going on, and we have weekly group meetings, journal clubs, and so on to try to keep up.
Skills do you feel are often overlooked yet essential for success, both in science overall and specifically in this area of immunology?
People, or let's say lay people, often think that there's a scientist and there's essentially a way how to do science. And I think it's actually very important to realize that science is a very individual approach. People are very different. People have different background. Like I'm, for instance, I'm German, originally German, and with being German, there comes certain mentality and approaches. And when I came to America, I saw, how Americans do research, and I see it here in Israel, how the Israeli-born people do it, which can be much braver. And I think it's very important to acknowledge the fact that there's not one approach to do it and not one strategy, but that we should actually take advantage of this heterogeneity.
Yes, that's super important advice. And what is the most valuable piece of career advice that you have ever received?
This was actually something which when I came to the Weizmann Institute, I talked to one of the senior PIs here, and he told me, you know, now that you start your own lab, and so you should make sure that everything which comes out of your lab keeps your standard and the integrity which you want to have it. And I think this is something very important, especially also in our current world, which is very fast living and let's say full of fake news and because of the media and other issues. I think it's very important that we personally set our standard. This is essentially what stays with you, right? You kind of have the possibility and the responsibility actually to make sure that your name is associated with high quality output. I found this very important, and I try to do this my best in this respect.
Is this also the advice that you give to others? For example, someone who's just starting in the research field, or do you have any other advice that you would like to share with us?
So one thing is, yes, I pass this advice on, and the second one is that I think I tell everybody to follow his or her intuition and to try to keep the eyes open, not to again not to follow the main path, but even dare to do some new things, at least at the site, and develop things and have a close look at data. That's what I mentioned before, that it's very important to look for the surprise findings in a way. This is what makes the fun. And there's always, I remember actually when I did my PhD, I presented my data. So this was one very nice thing when I was in Cologne. We had visitors from many very prolific scientists, and we got the chance as PhD students to present our data to these people. And I remember I presented my data, which I just got, and there was one outlier. And I just really said, okay, outlier. And I said, don't, I wanted almost to hide it. But this scientist, Max Cooper, actually made me aware to look at this outlier, and it developed into a very interesting side project in the end.
We would like to know also a bit more about your life outside of academia, since this is a mentorship kind of program. Like, how do you navigate the demand of high-profile scientific career with maintaining a healthy work-life balance?
I have family, so I have two boys, which actually now also both of them are studying biology. So it's somehow they got infected with the virus. So family, I think family is of course very nice as a balance keeper. I also like to read and listen to music, and so sometimes if everything comes too much, I think it's good to free the mind a bit. Work life balance is important, but it's also important to realize that this comes in waves. There are certain times where one has to give more than 100% of its time, and I think it's very important also to you know, let's say, have a partner which can appreciate this and knows that, this sometimes things are demanding. On the other hand, there's also very nice side aspects of science. And this is, for instance, the conferences, the exchange with the colleagues. And this is something which, it's not only, yes, we talk about science and we exchange data or facts, but it's also nice to see how other colleagues, handle it, and that they're under the same pressure. Right. One of the, maybe we haven't talked about that. Where are the frustrations or so? But I think one of lately, I think really problem in science is, you try to get the money, okay, you do the experiments and you get results, but the problem comes when you want to publish these data. And I think this is what's recently actually happening is very bad because it's very hard to publish in a reasonable time frame. And I think this is actually to the extent that it prevents scientific progress. And so when you think, we have now bioRxiv, we have actually the possibility to put preprints. And I just thought the other day, if we wouldn't have bioRxiv and these preprint servers, essentially science would be the progress would be prevented, because it's about these days 1 year, 2 years for major research points to get published and to reach the reader. And this is something I think the system has to do something about. It's very bad for careers because it's very hard for students to move on. And I think we have somehow, as a community, we have to try to find a better way to define essentially the bits which are to publish, right? Science is a continuum. You can always ask for more and you can always continue. But we have to start to understand that, it's about the communication, and we have to share our data. If it takes too much time, this is not good. It's a problem. So something which it's up to us actually to change this. I know many people think about it, but nobody has found a real solution, and it's also part of the domination of the journals actually and the publishing system, which is we have now for good and bad, which we probably should try to change.
Yeah, and this has for sure a huge impact on the life balance. That then with this frustration, you tend to work more, but then also you don't free your mind, as you were saying, and then the quality also is impacted now. Yeah, it's a huge issue. Have there been any sacrifices along your career path? Do you ever wish that you have chosen something different?
There's always something, I would have liked to work more on a host pathogen interface, and it just never happened. So I collaborate. I really cheer my collaborations with people which work in this area, and we haven't gotten beyond listeria, which is just it's not a real. I mean, for mice, not a real pathogen. Um, I think it's a very interesting area. So I still have some years to go. Maybe I will get there at some point. I think it's a very, very interesting area because many people have said this, that if you look at the host pathogen interface, now pathogens actually undermine the immune system. These are really interesting points, and I'm teaching this also in a course to see all these immunoevasins. Also, this is a very fascinating field, and of course, clinically highly relevant.
What challenges and which opportunities do you see for the next generation of scientists?
There is really an amazing development in the toolbox which we have. So we have concerning genetic engineering of mice, we can do now incredibly subtle modifications, which is very nice. So, for instance, we can recapitulate specific human mutations in mouse models and so on, which we couldn't do in the old days. Since I was involved in this early development of gene targeting and so on, I think people nowadays don't know how crude these approaches often were. First, it was very hard work because it was hours and hours to generate these animals. But the constructs also had a lot of limitations and were not optimal. Now with CRISPR-Cas9, we can do just really nice things, which means we have also to think much better for what are the experiments we want to do. And another huge opportunity because of this development is also to study human immunology, I think, which was much harder before. At least I think to study it in a relevant approach. So I think the future is really a combination of human immunology and mouse models, to run these in parallel and use each one to complement each other. And the advantage, or let's say disadvantage, of mouse models is that we work with these inbred strains, right, very genetically defined strains, with a limited spectrum of microbiota, and usually in a special pathogen-free conditions, hyperhygienic. And essentially, the human population gives us an advantage to look at the real life. And I think once this is done in a broader aspect, so that we look at large enough populations and so on, and again at the health situation, at the disease situation, at young, at old, and so I think there will be very, very interesting new findings coming. This is something which will be very exciting, and I think I would suggest people to try to link, let's say, basic research to these human focused studies.
And also this like human study and having large population with heterogeneity population, let's say, then it mean a lot of collaboration all worldwide to collect data, to look at it. So it is also something about take advantages of the network, the collaborative established is all part also of that, sharing, establish, collaborate.
Right, and it's also America where there are endemic parasites, where there's disease, completely different diseases, different problems. And I think this is something which is very, very interesting, and like a more holistic approach here is really warranted.
Yeah, it's a great suggestion for future generation. We'd love to hear a standout moment from your career, perhaps something light-hearted, surprising, out of the ordinary that you feel comfortable sharing with us.
There are these moments, it's like we said, experiments usually are hard. There's a lot of frustrations, but then there are these moments where you look through the microscope and you see a picture and this is a paper, or this is this is something I say wow, but I know my colleagues will also say wow. And so we had I had a few such moments actually in my career, but these are kind of things. So one of them, um, so we generate animals which have reporter genes. And so what we did is we did adoptive transfers of monocytes into animals where we had depleted macrophages. And the idea was to look for the progeny and the product of these monocytes, study monocyte differentiation into macrophages. And so what we did, more like almost just playing around, we transferred monocytes which were labeled red and were labeled green. And when we did this, what we saw is that the intestine was actually populated, the villi, the individual villi of the intestine were populated exclusively by red or green macrophages in the end. So what this showed was that there is a kind of seeded, the individual villi are seeded by individual monocytes, but that these monocytes have also an enormous potential to proliferate. And that was actually, back then we thought this is a bit of an artificial setting. So it was very striking. The picture was very striking, and we and others were very surprised. But we thought initially it's an artifact, but then Judy Allen has shown that indeed tissue macrophages can proliferate, and which was also quite a surprise. And so together with our finding, actually, this made a lot of sense. And I think this has become also an important factor which has been overlooked, also in relation, for instance, to Yamanaka and the D-differentiation. I think we have to become, the term terminally differentiated cell, I think one has to take in a much more flexible view. And again, in vivo, a lot of things can happen which are quite surprising. Yeah. So maybe this is not so lighthearted, but it was surprising.
Thank you very much for joining us, Steffen. It was a pleasure to talk to you. That's a wrap for today's episode of Climbing the Immune Ladder, Steps for Success in Science, iiSIAR podcast. A huge thank you to Steffen Jung for sharing his insights and experiences with us. We hope you found this conversation as inspiring and thought-provoking as we did. If you enjoyed the episode, don't forget to join us on the next episode and share it with anyone who might benefit from it. You can also follow us on our webpage to stay updated on future episodes and innate immunity updates. Thank you for listening, and remember, persistence and asking the right questions are what drive science, and maybe even your own journey. Until next time, stay curious.